So I'm going to select a telomere sequence from my list of genes, and then I'm going to sonify all 3 reading frames, and then I'll play it. It'll sound quite repetitive, which is the telomere repeats. But there'll be a change. There you go. Don't. So it's switched to a different melodic pattern because it's been put out of sync by one bass pair, which is a mutation.
It only takes one bass pair to knock that out because we're doing stuff in the reading frames, right?
So that big change in audio is caused by one bass pair change.
I can't think of any more human activity than conducting science experiments.
The game I play is a very interesting one. It's imagination in a tight straitjacket.
The beauty of a living thing is not the atoms that go into it, but the way those atoms are put together.
What I always think should be the basis of education is not answers but questions. We should teach kids how to question.
Okay, so we're here with Dr. Mark Temple. Actually had Mark as a lecturer for molecular medicine. Those are really fun. Those are really fun. Yeah, this is a few years back.
Had that pleasure.
So Mark is a molecular biologist. He's interested. I saw that looking at chromatin structure, cell cycle stage, aging.
Just dropped the real sciencey terms right off the bat.
I think you've just gone through my profile on the uni page and picked out the keywords. And there's lots of keywords there, but they include aging and
So that's the stuff for people who want to get into research or are interested in potentially collaborating with you, so they know broadly what you're working on.
In terms of students, I think any student who's engaged and enthusiastic is an asset, and I think any academic would want those kind of students. So regardless of your actual expertise as a student, if you've got those enthusiasm and engagement attributes, you should be able to talk to anyone, basically.
We really wanted to talk to you, me in particular, because we share a part of a story together. We're both musicians, actually.
So, strictly speaking, for a long time I was a drummer, and drummers are people who hang around musicians. That's the joke. So, but I would consider myself more musical than I previously thought. So, what do you play?
Quite a few instruments. At the moment, I teach guitar, and probably that's what I'm playing the most. But I played in bands playing bass and actually even a bit of saxophone. And the first instrument I learned was piano. Piano is a really good start instrument, I think.
So I've talked to a lot of academics, and quite a lot of them have done music in the past. And some of them are quite nostalgic about the music they did and never took it anywhere. But they're also good at science. So do you pursue what you enjoy, or do you pursue what you're good at? And if effectively you have to pursue what gives you an income. That's the bottom line. So I've always had a strong interest in music, and when I don't pursue music, I think I get depressed. Like if I don't play music for 3 years, like just say I'm going to focus on my PhD, or I'm going to focus on this, I can do that. And I can do well at science, but I find myself getting depressed. And when I re-engage with music, suddenly that part of my brain is back to life, and I'm doing science, and I'm doing music, and I'm in a good space. So I wouldn't give up music like a lot of people do. I think,
I totally agree. And it's like I still do a bit of private tuition, and the money's okay for that, but that's not why I'm doing it, really.
Maybe you can teach me a few things.
The reason why I'm doing it is because that's an outlet for me. I don't really have the time to go and play in bands and gig and things like that I did in my past, but being able to just privately teach is an outlet for a couple of hours a week, a few days a week, just to go and play some music with some people. It's great. But even things like this podcast, it's really good even to have just a creative outlet, right? Because this is video editing and things we're getting into now as a creative process. And I think balancing that with the more analytical side it's enjoyable, it gives you that break from the
Yeah, absolutely. I think when I go back to my burgeoning undergrad days, which was, we're talking early 1980s here, so a long time ago, I went to uni not playing music. I went to music as kind of like this, kind of partly depressed, kind of not know where I'm going kind of person. And I started doing an engineering subject and didn't like it, so I drifted into biology and rah, rah, rah. And I remember sitting in the back of a chemistry lecture with these two dudes who became good friends, but they were just dudes back then. It's like, who are you? And I'm sat with them. And then one guy, rather than listening to the lecturer, he was writing poetry. I was like, oh, that's interesting. And, I said, well, I'm trying to learn an instrument. You should, poetry is songs. We should do some stuff together. And he's like, yeah. And the other guy was his head around going, I play guitar. I thought, okay, let's form a band. Yeah. So in the back room of the chemistry lecture, we decided to do stuff together. Right. And the three of us in that lecture played together for years and then finally expanded to a 4-piece. And that 4-piece became the band that broke up and then reformed and became the Hummingbirds. Yeah. So it all came from, these bored students at the back of a chemistry lecture. I love chemistry, by the way, if you're listening, Janice and all you other chemistry lecturers. We couldn't live without chemistry. But sometimes you get distracted. Yeah. And we got distracted and formed a band. Yeah. And then I had my music, which was a real thing then, and my science. Yeah. And it was just a matter of juggling the two as best he could. Yeah. For a lot of people, it's not an issue because music doesn't go anywhere. So you don't have anything to manage. You've just got your science and you've got your little hobby. Yeah. But for us, the music went somewhere. So suddenly juggling the music activity and the science activity became difficult. And that's when I had to make a decision, do I do science or do I do music?
So were you like performing and doing like tours and stuff like that? So
well, it doesn't happen instantly. The first band with those friends in chemistry, when we got a fourth person in, we did 5 gigs in 2 years. So it was hardly busy. Yeah. And it was easy to manage that kind of stuff. Yeah. And then we broke up and I did honours. I did an honours in plant biochemistry. Go figure. And then after the honours, again, I hadn't done music for a year. I felt, I've got to do something, I really got to, So I called up my mates and said, look, let's have another go. And they were, yeah, all right. So we got back together and had another go. But we were much more focused because we had a year off and because we'd been in the band that didn't do very well, we knew we had to be more focused and professional about what we were doing. So we were very focused and professional. We were very much more song-based. We recorded our material, we promoted our material, and we tried to play shows. And then it just it pretty much from the first show, before we left the building, we got asked to play another show. And then we played the second show, and then we got asked to play a third show. And it just never stopped. From that point on, people just kept asking us to do stuff. Yeah. And we just said yes all the time. We always just went with it. And then it just ballooned out of control. Not out of control, but it became more successful than we anticipated. Yeah. And then that's when I had to say to my supervisor, because I was doing an MSc then, I can't come in on Saturday to do that thing you want me to do because I've got to go record. Yeah. And he was like, yeah, whatever, blah, blah, blah. And then 3 weeks later, when the recording date come, he goes, and by the way, you're coming on a Saturday, aren't you? And I'd be like, no, I'm recording. He was like, no, you're coming in on Saturday. Yeah. So I went in on Saturday really early and then bailed and was late to the recording. And I was really stressed out because I was just trying to do 2 things. And then that started to happen more and more frequently with the gigs and the early morning starts. And at some point I went, I can't do this. And then particularly when they say, can you come into the studio for 6 weeks? All day, every day. So it's like, well, I've got to give up science. Yeah. So we did. We all kind of gave up our day gigs and said, let's try this full time. Yeah.
So you gave up science at that stage.
I did.
And then to pursue the music. But now you're a scientist. So there's a disconnect here. There's something missing from your story that we need to hear.
Well, I gave up science. So the disconnect is, how did I leave the band and get back into science? Yeah. Because, if you think about it, if you've done music for 5 years or 6 years, my passport had professional musician as my occupation. I was very proud of that. It's like, oh, look at that. I'll show my friends, look, I'm a musician, And when I left the band, okay, for whatever reasons, it was one of the most difficult things I ever did. It was having to walk out of my buddies and say, look, I can't do this anymore. But I left and then went back to science and go to these interviews. And I was really stressing out because they're going to say, well, Mark, what have you been doing? What makes you good for this job? And I can say, well, I've been drinking lots and playing music in a rock and roll band. And so I was like, well, how are you going to get a job? Yeah. So I was really anxious, what do I do? Yeah. But
science moves quite fast as well, right? Like if you're out for 5 or 6 years, probably depends what field you're in largely, but you're probably missing quite a bit of latest research.
It does move quickly, and I think when you're young, you think that the information you have is what's going to get you the job rather than your ability to do things is what's going to get you the job. Yeah. So those bits of information you've learnt are what you want to be current. Mm-hmm. And they're going to get out of date within 5 years. So what I knew was kind of out of date, and I managed to talk to some old friends and get some tutoring work and some demonstrating work. And I did quite well at that and then got a job directly into a laboratory assistant position, which is not something I would have wanted to do. You don't study a master's degree in science to go and be a lab assistant. Yeah. So I made that decision that I think I need to come in at a lower level than I would have had I not done bands and music. Mm. And that worked. So I got a job as a lab assistant, and I got time to do my MSc part-time. So they gave me a little office next door to the lab. Mm. So at 4 or 5 o'clock, I would just jump into my office and do a bit of writing and finish off, a bit of analysis and stuff. And did that for 2 years. So that got me back into science, because once I got my MSc, I was then able to get a research assistant position. And then that gave me a bit more freedom and started to mix with the academics a lot more. And then, I was still in a part-time band at that stage. Because when I left, you don't need to know this as a science podcast, but when I left the band as a drummer, part of my reason was I wanted to do guitar vocals. I wanted to be, at least at some point in my life, front a band and sing to the audience and play my loud guitar and do a guitar solo and do the rock thing as a front person. I wanted to have that experience because I was always at the back. And being in a successful rock band as the drummer, to suddenly go, I want to sing and play guitar, was a big deal, because suddenly your first gig's going to be in front of 4,000 people or 3,000 people, because it's a big gig. Oh, yeah. So you can't do it on a little... So the band kind of, Simon at the time had to go up playing drums and I had played guitar, and we did a couple of my songs. And I listened back to them a couple of years ago and went, gee, they're awful. Yeah. But at least we went through the process of me having a go. Yeah. But I couldn't face the prospect of fronting that band for 1 or 2 songs in front of a big audience. I was just getting a bit nervous about that, and I thought, oh... So, and also I had no money. We were going through a financial thing and a big argument with the record company that was intractable, and I couldn't see how we were going to resolve it. So, like I said, you do what pays you an income. And I thought, well, this is intractable. I want to have a go at singing. Maybe I should try just leave, and had to get a job. So then I stumbled back into science.
It's a tough decision, isn't it? Because bands are very... they're very personal. You get very close relationships with the people you're in bands, I think.
You do.
It's more than just a normal professional relationship.
Divorced, I'm assuming.
It is. And I think when you're... Like we used to talk about cabin fever, because we were in each other's company all day, every day, day in, day out, for years, Yeah. So we used to say, you get to know more about a person than you ever imagined you would know about that person, who's not your partner, And... But on the other side of things, it's a lot of fun being in a band. It's really great fun. When it's working, it's really great fun. But when you're in a band, or when you're in any sort of situation, you don't think about leaving and how it's going to end. And you see it as being ongoing, and it's not going to end. We're going to keep rolling. And when this contract expires, we're going to get another contract. When the album's recorded, we'll record another album, and it'll go on and on. In the same way that when you're doing science, I'm going to get this job, I'm going to publish these papers, and I'll go somewhere else, and I'll work, and I'll publish papers, and it'll just roll on. But music's not as structured as science. Science is very structured. You can lay out a career path where I'm going to do undergrad, honours, PhD, get a postdoc, do a couple of postdoc positions, get a lecture position, and then I'll be right until I retire. And for a lot of people, that is a fairly easy career path to follow. And I stumbled into that career path as well. And I'm very grateful the fact that I managed to. I see my job as a lecturer as a second opportunity. Like, you know, I think often you only get one opportunity in life, and I think having the band opportunity and the science opportunity has been great for me. I feel very lucky in that respect. We've rambled on. I haven't quite got to the point, but I've almost forgotten what the point is.
The point, no, it's a conversation. It doesn't have to be a punchline. Cool. So I'm curious, how did you get into molecular biology? Because you said you did plant biochemistry, but
so there's two types of scientists in this world. Good ones and bad ones. No. There's two kind of scientists. I reckon there's those that focus early on and they manage to maintain their focus for their career. And the advantage of that is you become a world expert in something. And then that gives you that authority to be an expert and to go to conferences and talk and to lecture and do all that kind of stuff. I haven't followed that career path. And that's been, in a sense, it's been detrimental to me, but it's also, it's my path, so I accept it as the way I got to where I am. So I did my honours in plant biochemistry, and then one day walking down the corridor, once I'd finished my honours, I bumped into a lecturer who I'd written an essay on NMR spectroscopy in one of the exams. And there must have been so few essays written on that part of the subject that the lecturer remembered me. So he started talking to me. He was friendly to me because I'd written the essay on molecular biology.
That's a good tip to students, right? Try and stand out from the crowd.
It is. Write an esoteric essay on an obscure subject and you will impress your lecturer. That's right.
But make sure you do a good job.
Otherwise you'll stick out for the bad reasons,
So he offered me part-time work because I wanted to keep doing some science stuff. So I jumped from plant biochemistry to NMR structure of proteins in one fell swoop, which is fine because, you know, like I said, I was engaged, enthusiastic, and he employed me. So I learned to do NMR with him. And then when I finished NMR, I bailed into the band, then came back from the band to finish the NMR. So I finished my MSc in NMR when I left the band. And as you can imagine, being halfway through a degree with an academic and then bailing causes friction between you and the academic. So we were had quite a fractious relationship. And when I came back from the band, I wanted to re-engage with my MSc. And my lecturer said to me, my mentor said, no, that's not possible. That was 5 years ago. Things have moved on. I don't see how you could possibly finish what you did. So I went and complained to the head of school, and he said, don't worry, Mark, we'll set you up with another NMR spectroscopist. And then when my lecturer, my mentor, who'd moved to Melbourne, found out, he said, okay, fine, we'll work this out. So he flew me down to Melbourne. I stayed at his house, and that's how I engaged in re-engaging with that. So I jumped to NMR following plant biochemistry. And then when I finished my NMR, I'm not that good at maths. And I think there's lots of smart people do NMR work, nuclear magnetic resonance spectroscopy. That's what NMR stands for. And I think there's lots of very smart people. And I just thought, I can't compete with these guys because, you know, I know that they're really smart. And I was scratching my head. And also, it was a small field of science. And I got cold feet. And then another academic said to me, I've just got a grant, I've got a position in my lab part-time in molecular biology. And I thought, actually, molecular biology sounds good because it's a widespread thing. There's thousands and thousands of molecular biologists. Because molecular biology is that rudimentary technique that you learn. Everyone does PCR, everyone does agarose gels, everyone, you know, in science. So I thought that would be a really good thing. So even though I liked NMR, I just found it too cliquey and I didn't feel competent relative to my super duper smart friends. So I jumped to molecular biology whilst I was doing a band thing. And then the head of school came up to me and said, Mark, why aren't you doing a PhD? And I said, well, I didn't do that well in my honours, and I did band, and I did this. And he goes, no, you should apply for an APA scholarship, because I'd published a paper whilst I was in my doing molecular, and I published a paper in NMR. So he said, the papers will add to your track record, and I think that will get you above the mark. So I just went, oh, okay. And I went and had a chat with my boss, who was employing me to do molecular biology. And he said, that sounds great. He says, I'll support you. If you get your scholarship, I'll top you up. So you'll still get as much as you get as a part-time worker, but you'll have freedom. And that's an important thing that I often say to point out to the students, is that if you're doing a postgraduate degree, you're going to get people who expect you to take on that topic and to think about that topic and to choose the direction at which the topic takes. So you start to have ownership on what you do. Where if you're just doing assistant work, you're forever doing exactly what is laid out to you by your boss. You do what the protocol, and you repeat it, and you don't modify it, you don't change it, you don't develop it. But once you've got a postgraduate degree, or whilst you're doing a postgraduate degree, you're encouraged to take ownership and to think about it deeply and to make decisions that affect the outcome. And that was the big difference. That was what really drew me into the PhD, because as I was working as a research scientist, I still had to do the amount of routine jobs because that's what I was being paid for. Whereas as soon as I got my scholarship, it gave me freedom to do the science and to take those techniques and apply them. So that was a really big thing. So that's how I morphed into postgraduate in molecular biology from starting out in plant biochemistry.
It's interesting you say that, because I have a similar experience. I came, I would needed some money, right? Being a musician, I was poor. I needed a day job, so I was like,
What's wrong with cold pizza? That someone left behind the previous day.
I was sick of cold pizza. So I basically had to reach this crossroads, right? What do I do? And I decided to do a lab tech job. I did a TAFE degree to get me in lab tech. But the thing I didn't like about that job, and what a lot of people maybe don't realize about science jobs, is how routine they are. Every day I had the same set of jobs to do, I tick them off, and it's good in some respects. I guess some people kind of like that, right? They can keep their work separate. It's easy. They don't have to think too much about it. They go do their job, they get paid, and they come home. But it didn't really kind of inspire me. And I did the undergrad originally just to kind of, progress up the ladder, but then kind of got bitten by the research bug about halfway through, thinking, this is really great, like this freedom I have to investigate what I want. So it really resonates what you were saying.
And now you're balling because you got an APA scholarship as well.
I wouldn't call it balling, but yeah.
Come on, dude. It's tax-free.
I think whether it's science or music, it's like 5% inspiration and 95% perspiration. So if you're doing an assistant position, you don't have that 5% of inspiration. It's just all routine. And it's just a little bit of thinking about what you do and those making decisions about what you do, and then just doing all that application and repeating stuff. But it's that decision making that's important. I think if you're a creative person, or if you just want to take control over what you're doing.
I totally agree. Let's talk about, so we've talked about music, we've talked about science and how you got into it. Tell us how you've been able to marry those two.
Yes, that's the quandary. So the issue is music and science are in different realms. Particularly if you're paid to do molecular biology, there's no outlet within what you're doing to engage in music. And that's fine. I accept that, and I accepted that for years. But, at some point I was sitting in my office scratching my head thinking, is there an overlap that I can exploit? Because I think to do well at what you do, you need to use all the resources you have available to you. So I was thinking, I've got a knowledge of music, I've got a knowledge of like theory and playing, and I've got a knowledge of molecular biology and thinking experiments and doing experiments. And I was annoyed that there was no overlap. So I was doing music outside of hours, and I was doing molecular biology as my day job, which is fine. And I thought there's got to be an overlap, and I was scratching my head. And then I came across this, I actually went over to the Marx Institute because they were having an opening. And part of the opening, they were talking about this space they've got set up that had you know, you've got 5-point surround systems where you've got 5 speakers. Well, they were talking, I think it was like a 20-point surround system. They had 20 speakers, all at different angles and things, to create this rotating world of sound. And I thought, oh, that looks really interesting. I've got to go there. And I did a bit of research before I went there. And I started to read about sonification, this idea of using sound for data analysis. So I thought about it. I thought about molecular biology, and I thought, why not take a molecular biology concept and apply sonification to it, create audio using a biological concept. And I thought about, obviously I'm a DNA person, so I thought, well, I'll take DNA sequence and create audio from it. A quick look in the literature, you find there's plenty of examples of people taking the bases of DNA, which is like the G's, A's, T's, and C bases that just repeat
so these
endlessly.
Are the rungs of the DNA ladder.
These are the letters.
Yeah.
Yeah. So, so if you think about DNA, it's a long linear molecule, and it's almost an endless molecule, a linear molecule that goes on for billions of base pairs. And as you run down that linear molecule, you basically all you're doing is running across these bases. So you're running across G's, A's, T's, and C bases in different orders endlessly, okay? And people have taken the order of those bases and just mapped them to musical notes. And it's very easy to do. The most obvious thing to do is to say there are 4 bases, therefore I'll map them to 4 notes. And people have done that before. The next most obvious thing to do is to copy biology. So if you think about a cell and how a cell operates, it has a genome, which is the DNA, and it converts the information stored in the DNA into a protein sequence. And there's a complicated process called gene expression that takes the DNA sequence into a protein sequence. And that process, for undergraduates, it consists of transcription and translation. Okay, so I thought that process of gene expression, of making a protein sequence, is a good way, rather than making a linear string of amino acids, which is a protein, I'll make a linear string of notes, which is audio. And that's been done before. So I copied what had been done before. So I just took the codons. So rather than taking an A and mapping it to a musical note, and then a G, a T, and a C in the DNA, and mapping them to 4 musical notes, you take a codon, which is 3 bases, and you map.
These are kind of like, so the A's, G's, C's, and T's, they're like the letters. But DNA works, it puts those letters into words, but every single word is 3 letters long.
Yeah. So each word that's a perfect, that's the perfect analogy. So each base is a letter and a codon is a word. And the cell uses 3-letter words. So 3 bases go to 1 amino acid residue. So I did that as part of my sonification. And then if you want to get something that's publishable in science, you need to have something that's new, something that's a different way of thinking about it. So what hadn't been done is that people hadn't taken other aspects of that process of gene expression, such as using start codons and stop codons. So the beginning and end of genes, you have these particular words. So I took those words and in my coding, I use those words to start or stop the audio rather than to start or finish a protein. And then the other thing I did, which I thought was interesting, is that these 3-letter words can be read in different ways. So one sequence can be read 3 different ways in the cell. So why not take one sequence and create 3 different strings of audio from it? And then find the stops and start codons in one of those 3 reading frames, and then create interwoven passages of music, each of those 3 instruments, if you like, stopping and starting, depending on where the stop and start codons are.
That's cool.
So I did that as my, I thought it was cool, and I listened to the audio, and I thought it was interesting. Yeah. I can play you a bit.
I'd love
if
to
you
hear
like.
some audio in a second, but I just have some music nerd questions to ask. Right?
No, go for
I just
it.
have to sort out some things. So a reading frame. So we have these codons, right? And if we start, these words, right? If we start on the first letter of the word, or the second letter of the word, or the third letter, that's what you mean by
that
the
gives you 3 reading frames.
So correct me if I'm wrong, but you had 3 kind of instruments. Did you have a different instrument for each reading frame?
Yeah.
Awesome. And so you compile these 3 instruments, and they're both playing slightly different melodies because they're starting from a different reading frame.
Yes.
And then what
so
about
starting from different reading frame means that because it's like they make new words basically, right? Because
They do. They do.
Letter of the codon,
Yeah,
you're
yeah.
using 3.
So if
then
you
3,
have
then
20,
3,
if you have 20 letters, if you start from the first one or the second or the third, then you get new bunches of words.
So for example, at the beginning of a gene, typically you'd have an ATG codon. So the letters A, T, and G form the first word. But then you slide along to the second base. So not the A, T, G, but the T, G, something else. And then you slide along to the third letter, which is the G, and then you've got G, something else, something else.
Yeah.
So they're your 3 reading frames. So you slide along one base at a time, and each time, so that your sliding window is 3 base pairs long, and it moves at 1 base at a time. And that creates all possible reading frames. But in a cell, you read the 3 bases, and then you jump to the next 3 bases. You don't slide along, and then you jump to the next 3 bases. So the cell has to decide which of those positions do I start on the first base, the second base, or the third base before I then jump to the next 3. The cell has to make that decision. So its start and stop codons help make that decision. So, sorry,
so you had those 3 different reading frames or a different instrument each.
Yes.
That then you were mentioning before that you used the A, C, G, and T as 4 different notes, but
Yes.
being a musician, there's 12 different notes in music, and that's
yeah.
just in one octave.
That's
we can
right,
have multiple
that's right.
octaves. So, so here's how I would have done it. I would have probably taken the codons because there's more. If we take those 4 letters, we can make 20.
You can.
20 words, 21 words.
I
should know that. I feel bad for not knowing that.
I can give you a complete breakdown if you want, but it's a bit nerdy. But I'll try.
No. Let's get nerdy. Let's get nerdy. So rather than just if we limit ourselves to those letters, right, which you were mentioning before, we can only hit 4 musical notes.
That's
so
right.
It's not very flexible.
It's almost monotonic. It's
it's not even a minor pentatonic scale.
No, you can't get your 5th note. You're stuck on those 4 notes. So you can't. You could probably play Smoke on the Water, but you probably couldn't play, I don't know, another Indian, I don't know, whatever song you want to pick. You know.
So how did you organize that? So I would have probably taken those codons, right, and had maybe one codon is a particular note, and a different codon is a different note. And that's going to leave you with a few codons left over, though.
Yeah. So if you consider 3 letters making a codon, which is what the cell does, then all possible combinations of all codons gives you 64 options. So if you've got, say you start on an A, you've got A. So at each position you've got 4 options. So if you're just looking at one base, you've got 4 options. If you're looking at 2 bases, you've got 4 options in the second position. That gives you 16 possible options. So you've got AA, AT, AG, AC, TT, TA, TG, TC, and then you go through. It gives you 16 options. When you get to codons, if you go through all the possible permutations, you get 64 options. Now, a cell is, they say it's a degenerative genetic code. So only 20 options are used in the cell. So there's in some instances, you can have 6 combinations of codons, 6 different codons code for the same amino acid. So in musical terms, those 6 different codons code for the same note, if you're following the genetic code. So instead of mapping, So I did it systematically. I thought, because I'm doing a science paper here, I need to be systematic. So I wrote algorithms that sonify DNA using 1 base at a time, which gives you 4 notes. I then chose algorithms that do 2 bases at a time, that gives you 16 notes. And then I chose algorithms that do codons, 3 bases at a time, that give you 64 notes. Now, getting back to your idea of octaves and things, 64 notes is quite a big range, particularly if you're playing a scale and not being chromatic.
It's larger than probably a lot of instruments, isn't it? 64 notes?
Absolutely. So if you start at the first note on a guitar and you want to play 64 notes, after about, 30 notes or so, you're at the top of the guitar neck and you've got nowhere to go. You've run out of notes. And the same with on a piano. If you start at the bottom, you might get to the end of the piano if you're being chromatic. But if you're playing a scale, so out of the 12 semitones in a scale, you only play, say, 7 notes. So you run out of piano keys. Yeah. So I had to go at sonifying DNA using 64 notes, but I had the output from that sonification is chromatic. It's not in a scale. But when you switch to doing what the cell does, so getting back to what I was saying earlier, I was systematic. I did 1 note at a time for 4 notes, 2 notes, 2 basses at a time for 16 notes, and then I went codons, 3 basses at a time for 64, but then I also did it as the cell does it. So I did the, I mapped it to 20 notes, which is what the cell does. So and then I also got nerdy, and I thought, well, since there are 3 reading frames, I need to have algorithms that play 3 notes at a time, intersperse 3 notes over 3 instruments, rather than just reading the gene sequence, which it gets complicated. I can play you examples at some point to make things a bit clearer if you want.
Whenever you want it. We should just mention your really cool phone case as well. Hamid might not, due to his age, might not know what that is.
It's a cassette tape.
How dare you, Alex? I know you're old, but you're not that old.
I've even got a pen so we can fix up the tape inside if we need.
I mean, you'd have to fill it up with paper to record over it again.
So I don't know where the camera is here, and not that one. That's not that one. Is it? Where's the camera?
There's 3 cameras. So.
Yeah.
Okay. So here's the camera here. This is one right here.
Yeah.
All right. So.
Down a bit. That's good.
So part of the.
Yeah, that's good.
Okay.
Yeah.
So
really good.
part of the job as a molecular biologist was. Part of my job as a molecular biologist. Was dealing with large data sets. So doing some bioinformatics to do coding on large genome-wide and high-throughput genetics. So I thought, why don't I apply my coding skills to sonification? And I thought the best thing to do was have a tool that creates audio in real time. So that's what I tried to code. And finally, I've managed to code it. So that thing I was pointing at you is my result of coding that creates audio on the fly. Yeah. So you give it a sequence, you push go, and it generates the audio. Cool. So this is just a random sequence, if I can get my thing working. So this is just a random sequence, but no idea what it's going to sound like. But this is sonified using my tool. So let me get my tool to work. It's flipped around, there you go.
So that's just a random sequence.
Yes, it is. So, okay, so I can explain that to you.
Yeah.
So it started off playing da 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 da, and then it switched to da 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 da.
Yeah.
And then it went dun 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 like that,
Yeah.
and then it went back to da 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 da. Okay. So to me, that means something about the sequence, because when it was playing da 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 da, the triplets, that means that all 3 reading frames were being heard. So remember, we're saying that the codon, that 3 base pairs slides along one base pair to read the next one, and then it slides along one, and then it gets back to reading the previous reading frame again. So the 3 reading frames. So it was playing, all 3 reading frames were playing, but when it started to go to the doublet notes, that means there was a stop codon in one of those 3 reading frames, which stopped the audio in that reading frame.
Yeah.
So you're left with 2 options for a gene sequence, if you like.
Yeah.
So it was playing da-dun, 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 da-dun. And then it started playing dun, 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 1 note, which meant there was a stop codon in one of the reading frames that was actually playing.
Yeah.
So it left you with 1 note. So if you hear 1 note playing for an extended period of time, that's a good indicator that it's an open reading frame, which could be a gene sequence. And then it went back to da-da-da-da-da-da-da-da. So those 2 silent reading frames then came across start codons, which reinitiated the audio to then start playing da-da-da-da-da-da-da-da. So in its own right, the audio is never going to be enough to do proper sequence analysis. Yeah. But as a supplementary tool to visual inspection, I think it's quite useful.
Yeah, I would think that would be cool to just find patterns. So just transforming the data in this new audio version could give you a new way of just trying to recognize patterns that might have been missed by other methods.
Because there's a lot of things in DNA like repeat sequences and stuff like that, isn't there?
Yeah. So when people do sonification, they often sonify almost random data, like stock market data or things like that. So a lot of non-scientists, when they sonify DNA, they treat DNA as a random sequence. Where, because I'm a scientist, when I treat DNA, I think of it as information content. And how do you listen to and sonify that information content? So using stop and start codons in a certain way is important for generating the audio. There are many other ways of sonifying DNA to make it sound more musical. And I've avoided all of those. But my concession to music was to use traditional musical instruments as the sound and to put them into a scale so that it doesn't sound dissonant. So that was my concession to making it listenable.
But I guess that's an important point, right? Because lots of stuff in DNA is about repeat sequences and tandem repeats and things like this. Is how a lot of people identify genes and things that are of interest in DNA.
Yeah, you've just put me back on track.
Yeah.
Okay, thank you. So if you think about DNA as a random source, then the audio is going to sound random. So when I was sonifying the DNA, I looked for the low-hanging fruit. Where's the easiest thing to have a proof of principle that the sonification is useful? So because I'm sonifying triplets, because that's what the cell does, sonifying codons, then I found a sequence which is made up of 6 base pair repeat, which means that it's going to cycle through a melody because it's 6 base pairs long. Okay, if it was 5 base pairs long, it would take 20 bases to repeat, but because it's 6 base pairs long, it repeats every 6 base pairs. I'm confusing you. But anyway, so if you listen to repetitive DNA, and a large proportion of the human genome is repetitive DNA, except we're not that interested in it. But it's easier to sonify. So I chose to sonify these sequences at the end of DNA, which are called telomeres, telomere sequences. And it's just, I can't remember the sequence. Is it TTAGGG or ATTGGG? It's one of those.
That sounds about right.
It's about right. It's about right. And so we have that 6 base pair repeat at the ends of our chromosomes. All chromosomes got them. And when you sonify them, it gives you a melodic pattern that your ear can register, as opposed to sonifying other DNA sequences that almost sound random, and the ear doesn't get a fix on what it sounds like. So when you sonify a telomere sequence, you get this repetitive pattern, and when there's mutation in the telomere, you can hear it straight away and go, oh, I heard that mutation.
That's cool.
And then I looked for other examples where you can have repetitive sequences, such as, I think it's Huntington's disease. There's a CAG repeat, and you can have 40 CGA repeats. So suddenly you get these very melodic patterns occurring in some gene sequences. I can play you a telomere sequence if you like.
Yeah, that'd be awesome.
I'm not sure whether you'll be able to make head and a tail of it, but I'll have a go. So let me just go back here. Let's see if I can navigate my own tool. Okay, so DNA sonification. Okay, so I'm going to select a telomere sequence from my list of genes. Excuse me while I do this. Here you go, human telomeric sequence. Okay, and then I'm going to sonify all 3 reading frames. So, and then, okay, and I'm ignoring the stop and start codons on this one because, well, just because. And then I'll play it. It'll sound quite repetitive, and I don't know, and I'll talk over it if you like. Which is the telomere repeats, but there'll be a change. There you go. Don't. So it's switched to a different melodic pattern because it's been put out of sync by 1 base pair, which is a mutation.
It only takes 1 base pair to knock that out because we're doing stuff in the reading frames, right?
So that big change in audio is caused by 1 base pair change, and they'll switch back soon because there's another mutation. There you go. It's a second.
That's
So,
amazing.
so when you listen to repetitive sequences,
Yeah.
That's when the penny dropped to me. Yes, it's useful if it's a simple sequence. So I've proven to I've convinced myself that it's a useful method of analysis, and I've done a proof of principle paper. So the next thing is to sonify sequences in different ways that remove a lot of that randomness, because you can't pick changes in the randomness, so that you can pick the changes. So I've got some very clear ideas on how to do that, but I don't want to spill the beans just right now. But it's going to be my summer project, is
cool.
to recode a follow-up tool to pick changes in more complicated sequences. And I think that's the battle.
I think we're kind of.
I have one quick question, and then we'll probably end it, because we have gone on a while, haven't we? So I told you time will fly. It's a fascinating discussion. I'm just like appeasing my own interest here.
Yeah, go for it.
But
if you're not interested, you can't expect the audience to be interested.
One last question, I guess, is, I guess, comparing like some utility out of this is human genes are relatively similar, right? Except for the odd like single point mutation here and there. Like the human genes, the genes we have that work are pretty the same, right?
Absolutely.
So if you're analyzing genomes of people,
so if I compare my sequence to your sequence, then our sequences are the same.
Yeah.
So you can abstract away what's the same and sonify what's different.
Yeah.
Which is the next round.
So that'd be your basically the melody of your soul.
Yeah.
Well, I don't know. I mean,
there's no such thing as a soul, though, man.
I mean, if you want to have fun with this, I think that the fun thing to do is to reverse code the thing. So take a melody of a song you like, and convert that into a DNA sequence, and then search the genome to see if that sequence occurs.
What a great idea.
And then start to say, but then...
Jimi Hendrix is in my genome.
That's right, absolutely. And I think that's a lot of fun, but I think it will confuse the general public. Yeah. And it's almost fake news.
Yeah.
And I'm really torn between keeping the straight and narrow and keeping it very scientific and having fun with it. Yeah. And I want to do both, but I don't want to complicate things too much. And it's hard. I'm still not sure what to do.
I could see like an app maybe in the future where you upload a music MP3 onto it, and it gives you a sequence, and then it compares that. The person doesn't even have to see the sequence, right? It just compares it to your genome, and if there's enough similarity.
So it's interesting where...
Or maybe it could go across species. Some people might be similar to like bears and dogs and stuff.
Yeah. So just in terms of where this takes you, I know you want to wrap up, but so like I said, I published this nervously thinking, I want to be taken seriously. I don't want people to think I'm just creating random audio from, trying to make music. So I was very focused in mimicking a cellular process to generate audio, and I think it works, and I think hopefully there's a little demonstration that says it works. But then it took off on social media and for me it became a big deal, and I started to get lots of people asking questions and stuff, and someone called me up or contacted me and said, can you play this audio as music at a science event? So I did that, and then we did it again. So that's starting to become a bit of a thing. Right. And then I got an email last week from an academic in Scotland saying, can you send me some audio because I want to put it into Cubase, which is a music production tool, to generate audio. I was like, oh, yeah, okay. And then I did a search last week because I'm always trying to update to see if people are showing interest in what I've done. And I found out, and I didn't spot it last year because it was in Italian, but some Italian people had at least cited the idea of what I did and extended it and then done a musical performance in Italy about this thing.
That's awesome.
And so there's it's sort of little sparks here and there sort of coming through with taking this into a more creative domain. Yeah. And that, to me, says, well, I should stop thinking about taking it into a creative domain because I don't want it to make my science less, I don't want to confound the two, but I think I should just embrace the two. Maybe I should have a pseudonym for my musical exploration of DNA audio and have my science persona.
Yeah.
You know, like that Janus, that view where you've got your sort of your Harlequin, you've got your white face and your black face.
I love it.
Okay, cool. Well, thanks so much for your time, Mark. We really
That's
appreciate
all right.
it.
Thank you for showing interest.
That was a fun conversation, and when can the general public get access to those algorithms? Or
Yeah, it's online.
Yeah?
Part of the publication was the website.
Yep.
So it's one word, DNAsonification.com.